So, as we have discussed about the role of salicylic acid, I wanted to show you the pictorial representation of how a GPI anchor looks. Now, this is a picture which I just wanted to show you that how actually transmembrane proteins are made. When the ribosome synthesizes, there is two ways of protein processing. One is known as post-translational processing and one is known as co-translational processing. That is, when the protein or the polypeptide is being synthesized within the ribosome, simultaneously it is being inserted on the membrane. So as you can see, the protein is moving through the translocon and is gradually being inserted. The protein is moving through the translocon and is being gradually inserted and is being a part of the transmembrane. So this is how transmembrane proteins are made and folded. This is not a part of a syllabus. I just wanted to give you a glimpse of the picture of how transmembrane proteins are actually inserted inside the membrane. Now let's come to this portion. As I was talking about proteins, this is a continuation of the proteins, that most proteins are, as you can see, have carbohydrates attached to them. We have seen the structure of lipoproteins. We have seen the structure of glycoproteins. So from the outer surface, it seems that a carbohydrate layer, you can look at the blue circles, a carbohydrate layer is almost masking the plasma membrane. If you have a top view of the plasma membrane, it seems as if a blue carbohydrate layer is masking the plasma membrane. At times, this layer is called as the glycocalyx. The glycocalyx or the cell coat, which is specifically used to describe the carbohydrate-rich zone on the cell surface. So this question comes, what is glycocalyx? So glycocalyx is basically the carbohydrate-rich zone of the cell surface. As you can see, it's a blue masking done on the cell surface. Now moving on to the membrane dynamics. If we look into the two different states of the membrane, you can see that the membrane can exist in two formats. One is the ordered state, where the molecules are more in strict configuration. The bilayer cannot move within itself. In most cases, our normal physiological temperature is 20 to 40 degrees centigrade. So most saturated fatty acids, most long chain saturated fatty acids prefer to remain in this ordered state, ordered way, in a stricter configuration. But as the temperature rises and heat is gained by the membrane, you can see that the membrane fluidity increases and this state of the membrane is known as the liquid disordered state. Now what happens in case of unsaturated fatty acids? Let me show you the picture. Yeah, right, this one. Okay, so this is a structure for unsaturated fatty acids, and you can see that in case of the unsaturated fatty acids where there is a double bond, a kink means fold. You can see the fold, the kink is being observed. So if there is much quantity of unsaturated fatty acids in the membrane, even though the membrane wants to be in an ordered state, it fails to remain in an ordered state. It thus becomes a disordered state. So if there is saturated fatty acids, no problem. We have, where are the phospholipids? Straight chains. But if there are unsaturated fatty acids, there are kinks. And these kinks create problems. So let me go back to that picture. So in case of saturated fatty acids, at normal physiological temperature of 20 to 40 degrees centigrades, the lipids are in ordered state. But in case of unsaturated fatty acids, in the normal physiological temperature of 20 to 40 degrees centigrade, just now I showed you, the presence of the kinks makes the lipid molecules difficult to be uh, in such a strict configuration, and thus the lipid molecules get themselves arranged in a disordered state. The LO, or the liquid order state, is also known as the gel phase of the plasma membrane. Mind it, in the order state, the mobility within the plasma membrane is totally restricted. But in the liquid disorder state, the molecules can move within the plane of the membrane. Now coming to the movement through the membrane. So let's go, I start with lipids because the lipid is the actual skeleton of the plasma membrane. So if we look into the three kinds of movement, the picture is given very beautifully. This is Leninger. You can see that there are three categories of enzyme. The first one is flippase. You can see flippase. Now locate, this is a phospholipid, okay? Now, what does flippase does? It catalyzes the translocation of the aminophospholipid phosphatidyl ethanol amine and phosphatidyl serine. Both phosphatidyl ethanol amine and phosphatidyl serine from the extracellular to the cytosolic leaflet. So at times it catalyzes the movement of PE, phosphatidyl ethanolamine, and phosphatidyl serine from the outer to the cytosolic leaflet. So let us divide the leaflets. If this is the bilayer, we can say that this layer, which is associated with the outer surface, is the outer layer, and this layer, which is associated with the inner cytoplasmic surface, is the cytosolic leaflet. So flippase is basically an ATPase. As you can see, it uses the energy derived from the breakdown of ATP to catalyze the movement of phosphatidyl ethanolamine from the outer leaflet, not only phosphatidyl ethanolamine, phosphatidyl serine, from the outer leaflet to the inner leaflet is it catalyzes that. Let me give you one physiological role of this enzyme. As you see that I had told you just in my previous class that phosphatidyl serine externalization is a hallmark of apoptosis. So if by mistake a phosphatidyl serine has moved to the outer leaflet, then flippase takes care that the phosphatidyl serine is brought back again to the inner leaflet. So this function of flippase becomes very important to ensure that phosphatidyl serine always remains on the cytosolic leaflet. Next, moving to the next category of enzyme that helps in the lipid movement is the floppase. As you can see, this is also an ATPase, that is, it is using the breakdown energy of ATP to transfer a phospholipid from the cytosolic leaflet to the outer leaflet. So flippase and floppase act together, they're partners. And next, scramblase. This is a more efficient one. This uses calcium. Scramblase activity is associated with the rise in cytosolic calcium. Okay, so rise in cytosolic calcium usually occurs when there is either apoptosis or there is a cell injury. So this is important. So since it is associated with calcium, so high level of cytosolic calcium is an indication that there has been an injury or the death process has started. And then only what scramblase does, it at times moves the proteins of the inner, the phospholipids of the inner leaflet to the outer leaflet. So that is scramblase. It can move lipid in both directions. This is Another picture you can see flipase, flopase, scramblase. And this is the movement. As you can see, the flip-flop movement, you can see the motions as per second. 10 to the power minus to 4 to 10 to the minus 3 per second. Then we have the rotational diffusion movement. The keeping the head constant, the tails can move, it has been shown. So there are certain rotational diffusion movement observed in the bilayer. And as I said again and again, given the fluid nature of the membrane, the lipid molecules can move laterally. So there is a lateral diffusion movement in the membrane. So this is the rate of the lateral diffusion movement. It's 10 to the power 7 per second, and the rotational is 10 to the power 8 per second. Okay. So, and then some bond vibrations are also seen in the fatty acid tails of lipid molecules. I'm not going to go into the details of that. I just wanted to show you that different kinds of movements and their rate, the time. So these are the variety of movements. And we studied the enzymes associated with the movements, specifically when the lipid molecules or the phospholipid molecules moves from the outer leaflet to the inner leaflet, or from the inner to the outer, or both ways. Now, coming to the experiment, this is a question that comes, that how would you prove that lipids and proteins diffuse in the bilayer? I have said that they move, and Singer-Nicholson also proved and said that they moved. That is the fluidity of the membrane. So experimentally, how this was proved? Let's look into this experiment. So this is a cell. And you can see at the first instance, the lipid molecules of the outer leaflet are attached to fluorescent molecules. So you can see the head of the lipid molecules are attached to the fluorescent molecules. And as a result of the attachment of the fluorescent molecules, look into the red semicircle, the red boundary. So the whole membrane has been bounded by a layer of fluorescent molecules. You can see the, you can see the red boundary. Now what happens? A laser beam is applied to one part of the membrane. When a laser beam is applied to one part of the membrane, what does the laser does? It bleaches out the fluorescent molecules. You can see a white portion is being created. Look at this. So this was the colored boundary. We are seeing that the membrane is covered by fluorescent molecules, and then a laser beam is being forced on the membrane, and the point where the beam hits, you can see that the fluorescent molecules have been bleached, so they are not showing their colors. Now let us imagine, if there was no movement in the lipid molecules, then this bleaching would have remained as it is. It would have been fixed at a point. But since there is movement, now and again I have constantly said that there is movement within the lipid bilayer, so fluorescent molecules from the lipid molecules bearing fluorescence from the adjacent areas move to these areas. You can see the lipid molecules bearing fluorescence from the adjacent areas are moving into the bleached areas, and this area is again giving a fluorescent color. So this experiment was a very efficient experiment to prove that lipid molecules are usually diffuse greatly through the bilayer. This experiment was known as a fluorescence recovery after photo bleaching, or FRAP method. So this experiment comes, and this is just a basic table to show you the difference of the membrane in between bacteria, yeast, and higher organisms. And these are the molecular shapes of the lipid molecules. This is just a picture. And I'm moving on to an area which is not a part of your syllabus, but I thought of touching it. That is the model of a raft domain. Now imagine that there is a pond and certain logs are dispersed in different areas of the pond. There is a pond and small floating logs have been dispersed on the surface of the pond. Now, whenever a signal is coming to the pond, the log starts moving towards each other to form a raft. So, if we think of that picture and superimpose it on a membrane, so imagine that the receptors are dispersed on the membrane. Whenever a ligand molecule moves near the membrane, the receptors move through the plane of the membrane to get aggregated at a place. These local aggregations involve a lot of chemical networks, a lot of chemical interactions. So these local aggregations are known as raft. As you can see, the molecules have aggregated. So just, you don't have to study this in much details. I'm showing another, see. So these are rafts. The receptors have aggregated. So this is specifically a raft, and it has been seen that in the raft areas, the GPI anchored proteins are maximal. It has been seen, it has been studied. Let me show you another picture. You can see the raft accumulation. So these are rafts. So when there is a signaling molecule or the receptors move together, they form small rafts, which helps them to connect with the signal in a strong manner. So initially they are dispersed and they aggregate to form rafts. So this is just a pictorial representation of raft. It's no point in getting into much detail about this. Now, I showed you one experimental evidence which proved that protein and lipid molecules in the membrane can move. There were further experimental approaches which showed that there are two categories of protein. One is the peripheral and the other is the integral. So this experiment was known as the freeze fracture technique of the plasma membrane. What happened in the freeze fracture electron microscopy of the plasma membrane that was done by Branton 1968 via a very sharp blade. The lipid bilayer was dissected and then liquid nitrogen was poured through the bilayer and the plates were studied, and it was seen that where there were integral proteins, there were holes. In one layer, as you can see, the integral protein has moved to that part, so there were holes. While in the other layer, the plate gave the picture of protrusions. So this was the famous freeze fracture technique of Branton, where Branton showed that the lipid membrane is made up of two categories of proteins, the peripheral protein and the integral protein. And this is another example of the lateral movement of protein and lipids. If this question comes, you people can give the Frapp experiment, or people can give the famous experiment of Fry and Edin in 1970. What did they do? Look at the picture. This is a mouse cell. This is a human cell. Now, there are membrane proteins on the mouse cell. There are membrane proteins on the human cell. The first step of this experiment was that the mouse cell and the human cell were fused to produce a heterokaryon, a hybrid cell. Now, the mouse proteins were labeled by one color, say green color, and green color fluorescence, and the human proteins were labeled by a red color fluorescence. And time was 0 minute, and the cells were incubated at 37 degrees centigrade. So we are starting with the cell, on one half of which it has red color and the other half of which has green color. But after 40 minutes of the experiment, it was seen that a mixture of red and green color was visible throughout the membrane, indicating that both the mouse proteins and the human proteins had moved laterally along the plane of the membrane. So the green fluorescence that was used was basically fluorescein, and the red fluorescence that was used was basically the rhodamine. Okay, so this was another experiment to prove the lateral diffusion of the proteins and the lipid molecules. I mean, this was a lateral diffusion of the proteins. So if the question comes, prove by an example the lateral diffusion of the lipids, give the FRAP experiment. Okay, when it comes as the lateral diffusion of the lipids, give the FRAP experiment, because this labeling was done on the lipid heads. So it was basically an experiment to see the lipid movement through the bilayer. Okay, and if the experiment question comes that give an experiment to prove the lateral movement through the proteins, then specifically give this one, because the proteins were tagged with fluorescent probes. And if the question comes that through an experiment show that lipid and protein movement takes place, then you have a choice, you can give either, anyone. So depending on the questions, give your answer. So today we'll be doing till this much. I'm not moving into the junctions today. So I'll be taking questions regarding what I have told now. So we have finished the structural part of the plasma membrane.